log into? I have a separate question for you guys. Are you all getting emails about setting up dual authorization for Scholastica, or is that just me? Okay, great. I don't want to do it, which is why I'm asking. I usually have my phone with me, but what if I don't? Okay, so I'm going to start making my own doodles up here. Hopefully they look similar to what you and your neighbor have. What should we do? Up at the top. Try that. Okay. So hopefully what we got, right, is some type of polar head. It's going to include some phosphate, but you know, this is biology, so it's fine if you don't write the actual atoms out. Then we're going to see two fatty acid tails. Okay. Next, someone remind me, what kind of properties does this polar phosphate head have? Yeah. So hydrophilic. And what about the fatty acid tails? Yeah, exactly. So I have these hydrophobic water field. But they can't really be abbreviated because they both be HP, right? Okay. So what is this going to mean for water? Where would we see water clustering around this phospholipid? Yep. So here's our water. And if we were to have another phospholipid, where do we think it would go? Would it go over here on the left? Would it go over there on the right? Okay, So what am I going to draw first if I start drawing here? The head or the tails? Yep, great. We got the tails here. And this is going to behave exactly the same way. This is still going to be hydrophilic on the head, hydrophobic on the tails. So when we have water, it's going to orient over here as well. So this is what we'll see when we get to our actual cell. So we're not going to expect to see water anywhere in the middle here. No water here. Awesome. Okay. So a few items of course business before we get into our new stuff. I think you all got a reminder email, but reminder now, SI starts tonight. So the way SI works is basically your lecture TAs, not here physically, but this is supposed to correspond to lecture. They have practice problems, worksheets that they distribute to you. You can go in, check with them, go over them. It's also a great time just to kind of ask questions about the material we're going over in lecture. You don't have to just, compare your answers and then leave. You are welcome to do that if you're feeling nervous, but it's helpful to get some personal interaction, right? That's part of the reason we have these many SI sessions, review hours, office hours. It's really to give you some more personal kind of time with class goes. And our section is small, but still a lot of lecture to get through. Okay. For those of you who may still be waiting on books, we're standing in the chapters literally right now, so they'll be up this afternoon, which is great because mastering A&P, for those of you who haven't done that already, is due on Friday. So make sure we have that done right. Yeah.
So we did the trial, and it's gonna end soon. What do you want us to do with that?
Yeah, hopefully your books and access codes will get there by the time you do the next one. I don't remember what the date is for chapter 3, but if it's not ready, we'll figure it out when we get there. Basically, I'm gonna hope that the bookstore gets their act together. Another question to make sure you guys understand, because I know there's a little confusion about the lab bright spaces and where you were supposed to be for lab. So I'm actually just going to show you. I put an announcement in the correct lab brightspace course. So if you go to your lab brightspace course and under announcements, there isn't a note from me saying this is the right one, then it's not the right one. But in terms of finding where you should be, posted both in that course and in our lecture brightspace here. So I'm in our lecture brightspace. So under course resources, you have a lab schedule. Okay. So these letters correspond to whatever you signed up for in Banner. The merged lab course says Lab A on it. I don't have any powers to change that. I changed the title when you clicked into it, but it's still going to say that on your course landing page, and I can't fix that, unfortunately. But the way you read this, right, your lab letter, find the date, and it's going to tell you whether you're in anatomy or physiology. So what is today? The 15th, so our next labs are on the 16th. So if you're lab H, you'll be in phys. If you're lab I, you'll be in anatomy, and then the following week you'll switch. The lab schedule also has the times down at the bottom in case you forget. And as we get moving forward, it tells you a little more about what you're doing, right? So we also have the lab practical listed here at the end. Okay. Does anybody have any further lab questions or confusions? I feel like that's one of the difficult organizational points for this class. So just for those of you who have already had lab, just remember that you'll have a quiz at the beginning of your lab in 2 weeks, right? But next week you'll just be in either anatomy or phys, whichever one you didn't have yet. Okay. So when we left off, we talked about our carbohydrates, which are polar, characterized by having a lot of OH groups. They have this 1 to 2 to 1 carbon, hydrogen, and oxygen ratio, but basically all we're saying is there's a lot of oxygen. We talked about, what was the second group we talked about? Oh, and we had started our lipids, right? We started talking about lipids. We talked about the phospholipids, which are really important for forming our cell membrane. We also looked at the structure of a triglyceride, which is like your stereotypical lipid. That's what fats, oils are. Has 3 fatty acid tails bound together with a glycerol fascicle. So we do have a couple rarer forms of lipids. Again, they're going to be linked with this group based on the fact that they just have a lot of carbon and hydrogen, and they're therefore non-polar. So they're going to try to escape water. They're going to be hydrophobic. So these eicosanoids are a rarer category of lipid for us. They're basically a modified fatty acid that does play a role when cells are communicating with each other. We won't really talk about this group as a whole again in the future, but we will see some examples of them when we talk about signaling. That'll kind of come in when we are thinking about hormones and different physiological processes. So prostaglandins are an example, thromboxanes are an example. Steroids, we will see a fair bit. So all steroids are derived from cholesterol. So they all have the same ring structure. So we have these 4 rings and pressed up against each other. So we have seen some ring structures before. So does somebody want to tell me how you can tell that this is not a carbohydrate? Because we saw some rings when we were looking at those mono, disaccharides, polysaccharides. How can we tell that this is not one of those? What don't you see here? Yeah, exactly. You don't see oxygen, and when we're looking at a carbohydrate, these rings wouldn't be bound together at the 2 points. They'd be separated with an oxygen covalently bound to each ring, kind of to either side. So they'd be spaced apart. So because all steroids are derived from cholesterol, all our steroids are going to have this characteristic ring structure, and then they'll have stuff bound off the side, basically. So if you see these 4 rings, you're looking at steroids, but there will be some additional variation kind of at the side. So this is the part you want to keep in on. Cholesterol is going to be a key part of our cell membranes. We'll talk about why that is and what it does there as we move into membranes. But we'll also see that some of our hormones are going to be steroids. When we talk about hormones, they're basically some lipid hormones, so steroids, and there are going to be some protein hormones. So you'll be thinking about lipids or proteins when we think about hormones. But some pretty familiar examples like estrogen and testosterone are sex hormones. Those are steroid hormones, so they're lipids. Aldosterone is another one we'll talk about in future units. Any questions about lipids before we move on to proteins? Cool. Non-polar, just a bunch of carbon and hydrogen. The proteins are really important in physiology because they basically do everything. They're the real workhorse in physiology. And the reason for that is they form these really long polymer chains that can then, through a series of folding steps, produce a lot of different shapes. And when we think about function, it's actually the shape of things that gives them function, like chemically, biologically, right? You think about a person, right? It's important that we have legs so we can walk, right? If you think about protein, its shape likewise is going to determine what it can and can't do. So protein is made up of chains of specifically amino acids. So there's going to be a lot of variation in the proteins as well because there are a lot of amino acids. There are 20 different kinds, in fact. We're not going to memorize all the different 20. There are tables, you could look them up when you need them. but these 20 amino acids are going to be the building blocks of proteins. And it is important to know that different amino acids have different physical and chemical properties. So depending on what we call the R group, an amino acid could be polar, it could be non-polar, or have different behaviors, which means that different proteins, when they fold, because they're so big, may have a section that's polar, a section that's non-polar, maybe a section, I don't know, but they have a lot of different sections that have different behaviors and interact differently with different chemicals. So the bonds between our amino acids are called peptide bonds. Anytime you see that sort of root pep, you should be thinking about protein. So we have these peptide bonds, and if we have like a small little chain of amino acids, we'll usually just refer to it as a peptide. So that would be like 2 to 50, that would count as kind of small. When we're talking about a protein, we mean something big, potentially really big. So we're going to think about something that's more than 50 amino acids. Yeah. And you don't have to memorize those numbers, 2 to 50 versus more than 50, just this idea that they're both chains of amino acids, but proteins are super big. So when we see this peptide bond forming, when we look at an amino acid, it has a typical structure. So it has nitrogen on one end that's composing our amino group. So that's nitrogen bound to hydrogen. On the other end of the amino acid, we'll have a carboxyl group. So we have the carbon, oxygen, and our hydroxyl group here. Right, so those are parts of all amino acids, as is this little hydrogen on the bottom. The part we see varying here is this R group up at the top. So this says R1, so basically the same, adding a bunch of stuff. R2 would be a bunch of different stuff depending on where the amino acids are coming from. Okay, so the bond is going to form between the carboxyl group and the amino group. So when we're starting to look at a peptide, eventually a polypeptide, eventually a protein, we're going to see that if we look at the backbone, it'll always go in this specific order. We'll always have nitrogen, carbon, carbon, nitrogen, carbon, carbon, and so on. Maybe I'll even highlight the nitrogens in a different color, right? So this is going to be different than our previous groups that we saw, right? Than our carbohydrates and our lipids. Somebody notice what backbone looks like for the carbohydrates and the lipids? What if we had a line of covalently bound atoms together? What was bound together? What was forming that structure? Like when we do those squiggles, right? What am I drawing? Yeah, exactly. So actually when we draw a squiggle, every point is a carbon, right? And what you are supposed to just assume from the rest of the squiggle is that if nothing else is written on there, that the rest of those carbon bonds are filled with hydrogens, like that, and so on. So what those squiggles are those hydrocarbon chains. And if we saw a carbohydrate in a linear form, which we don't usually in water, it would similarly have chains of carbon. So if you see nitrogen mixed into that chain, you're looking at a protein. Good. So proteins have levels of structure. This is probably easiest when you're studying to just kind of make a little table for yourself. You have a bunch of slides on it, but it's not that bad if you look at it in a table form. So the primary structure of a protein is just those initial peptide bonds. So those covalent bonds between our amino acids as we form our peptide, and that's what we're going to call it as it gets longer, a polypeptide, poly just meaning many. Those peptide bonds are forming our primary structure. Our secondary structure is when things start to get more interesting. So in order to have these different shapes, right? These proteins aren't going to stay as these long polypeptide chains. They're going to start to fold and twist and get crumpled up. And chemistry, this is actually a pretty cool problem. They spend a lot of time trying to solve how proteins will fold. It's an active area of research. I think someone came up with a really good algorithm recently, and it was like in the news. I didn't look for the papers, but they used to have games where they would crowdsource how this protein folds. So it's actually kind of a hard problem, which is why we care about these levels of folding. But the secondary structure is going to have basically two choices in different sections of our protein. It's either going to form something called an alpha helix. So a helix is being kind of a curl, right, a swirl. Or we're going to form something called a beta pleated sheet. So pleated, just like a pleated skirt. It's just going to be kind of, it's going to be a sheet, so it's going to be kind of flat, but it's going to have these kind of zigzags in it horizontally. Does that make sense? Okay. So these two shapes are both formed through hydrogen bonding. So because oxygen kind of attracts more electrons, it has these slight negative charges on it. That's what formed our polarity in other molecules. It means that hydrogens get attracted to oxygen. So that's what we have happening here when we're looking at the secondary structure. So when you see these dotted lines, right, between our hydrogen and our oxygen, right? So hydrogen and oxygen, that's what's pulling these shapes together. And our tertiary structure, tertiary just meaning third, and we actually just name these 1, 2, 3, 4. Our tertiary structure is when we start to have more crumpling. So this is going to be less predictable. We're not going to have a number of like different named shapes that our tertiary structure is forming. We're just going to be taking these polypeptides that have folded into alpha helices and beta pleated sheets, and they're going to kind of crumple like you're scrunching them up like a ball of paper. This folding here is affected by the relationships between different parts of the protein, different R groups. But more specific than that, we can't really get because there's so much variation. Finally, when we look at a protein that's having action in the body, it's often not just one chain. Proteins often have subunits. So our quaternary structure, quaternary just meaning fourth, is the interaction of these different subunits. So what we have here is a picture of hemoglobin, which is a protein that allows your red blood cells to carry oxygen. So what we're looking at is that actually this protein hemoglobin has 4 different parts, 4 different subunits, which is why they're slightly different colors. Right, so here's one subunit, here's a second subunit, and then in the back we have kind of the same thing. We have a third subunit, and here we have a 4th subunit. So likewise, these subunits are attracted to each other based on kind of what properties the parts on that outside of that crumpled ball from the tertiary structure have. So depending on what R groups are sticking out on the outside of our subunit, that's going to control which subunits are attracted to each other and kind of what shape they make. Questions about the formation of a protein with these shapes. Your table would look something right. Okay. Type tool here, right? So our primary is our peptide bonds. Our secondary structure is formed by our hydrogen bonds. It's forming our alpha helix or our beta pleated sheet. Longer. Our tertiary structure crumples. Our bond here that we're interested in is those R groups. And our quaternary structure, this last one, is based on those subunits being attracted to each other. Yeah.
R groups and tertiary are attracted to each other, is that what you said?
Yeah, it's those
The,
R groups.
Of the amino acids.
Yeah, yeah, exactly. So like, I wonder if you can see it in one of the pictures there. It's not great, but like, basically as we start to have things pulling together, we'll have different R groups getting near each other. Right? Like this R group is getting kind of near that R group, so that might kind of pull them together and then form that tertiary structure after that. So let's do a couple of polling questions. And take another second or two. Take a look what we've got. Okay. Yeah, so it's our secondary structure. That hydrogen bonding is responsible for forming those alpha helices and beta pleated sheets. Our primary structure was those peptide bonds, so just forming that chain in the first place. Forming that chain like counts as one of these levels of the structure. Okay, so that brings us to our last group of biomolecules of macromolecules, nucleic acids. So just to count our four, right? We had carbohydrates, we had lipids, we had proteins, now we have our nucleic acids. So our nucleic acids are going to be based on these nucleotide monomers. So the monomer, the sort of smallest group that we'll have in a nucleic acid is a nucleotide. So the structure of our nucleotide is going to have three different parts. So we have, let's actually start with this base part, or sorry, this carbohydrate part, I should say. So we're going to have a ring structure kind of in the center of our nucleotide, which is then going to have these branches off of it. And think about it as like somebody was holding out a pom-pom like this. That's our nucleic acid. So that carbohydrate is going to be a 5-carbon sugar. So don't get tripped up by the fact that you do see these hydroxyl groups in here. That's normal for a nucleic acid. These additional branches that make it its own group. So we're going to have either ribose or deoxyribose here as our sugar in the center. And we see pictures of the deoxyribose versus the ribose at the bottom here. So somebody want to tell me what's different between these two rings? Here's the difference. Exactly. Yeah, there's an extra oxygen in the ribose. So we're looking down here. See this hydroxyl group, we see this hydroxyl group. If we look over to deoxyribose, this one's the same, but here we're missing an oxygen. So that is why it is called deoxyribose, right? So the name actually tells you D, like deleted, oxy for our oxygen, and then ribose because it's like the ribose. So deoxyribose is the one that has one less oxygen on it. This is what's going to give us DNA versus RNA. So the DNA is for deoxyribose, deoxyribonucleic acid, versus the R for ribose. It's going to give us our ribonucleic acid. So what we see coming off the carbohydrate, we got two things. First, we have our phosphate group, and this is going to be the same, both DNA and RNA. We'll have our phosphate group here. Okay. So we see a phosphorus, a bunch of oxygens around it, a phosphate group. On the other side, we're going to see our base. Okay. So the base here, not labeled as a specific base, but we have a bunch of choices. So those are blown up on the right-hand side here. Okay. So we have a couple of different options. We have the pyrimidines, which are cytosine, thymine, and uracil. We have our purines, which are adenine and guanine. And they're basically going to pair up against each other. What we want to notice as a difference between DNA and RNA specifically is uracil. Okay, uracil is only an RNA. Thymine is only going to be found in DNA. If we were to look at a strand of something, if we saw a bunch of U's in it, we'd know it's RNA. If we saw a bunch of T's in it, we'd know it's DNA. Something else that we can group together in this kind of nucleic acid group is a molecule we're going to talk about a bunch as we move into metabolism. So we have these kind of specialized forms of nucleotides that have, in our most important case, which is our AMP, ADP, ATP, right? ATP, we should recognize from our previous courses as our form of cellular energy, right? So energy. So what are adenosine mono, di, or triphosphate being our energized form is adenine, one of our nucleotides, with a additional 2 phosphate groups on it, right? So our adenosine monophosphate would be like that. Our adenosine diphosphate, yes, a little bigger. We have 2 phosphate groups, be like that. And then this full structure as drawn on the board here has 3 phosphate groups. So that tells you it's adenosine triphosphate. So that's our ATP. We'll also see 2 other sort of modified nucleotides. As we're doing metabolism, we'll see this nicotinamide adenine dinucleotide, or NAD. We'll talk about the role that that plays in basically ferrying around electrons in cellular respiration. We'll also see this molecule FAD, flavin adenine dinucleotide. And that's going to have a similar role as NAD as we move into our unit on the top. When we're talking about pathways inside cells, we'll see something called cyclic nucleotides. And specifically, what you'll see a lot is cyclic AMP. So this is really the one to remember, just for when you see it later. As you're thinking about what kind of a molecule is that? When we see cyclic AMP again, that's this modified nucleotide. So it's kind of a nucleic acid. What they do is they form a ring with a phosphate group. So instead of having that phosphate with just the oxygens around it here, it's formed this weird ring thing. That's where the C comes from, that cyclic name. Questions about those big groups of enzymes, or sorry, the big groups of macromolecules? Those are 4 classes of macromolecules. I'll do some quick poll questions after we get through enzymes. So be like 30 seconds per question. I'll look at the clock. Awesome. Yeah, the atoms we see in our biomolecules or macromolecules are carbon, hydrogen, oxygen, and nitrogen. Those are the main ones. We've seen, I mean, we might add phosphorus to that list depending on what specific macromolecule we're talking about. If we add in R groups, we might see some rarer things, but these form the basis of our macromolecules. Okay. What are our basic types? Yeah, so our carbohydrates, lipids, proteins, nucleotides, or nucleic acids. Which of these would not be a biomolecule? So now we're not thinking about classes. So some of these would be examples of specific biomolecules. So this is actually a little bit of a toughie because we don't really care about wax, so we don't talk about it much. But wax is technically a lipid. The molecule that is not a biomolecule is water. So water is what we're interested in interacting with our biomolecules, but like water isn't a carb, water isn't a lipid, water isn't a protein, and water isn't a nucleic acid. So that's how you kind of remember that, right? Water is just H2O. It doesn't have any of the characteristics of these big classes of macromolecules. So it's important for our cells, but not a biomolecule. thinking about polar molecules. Really, for all our molecules, we're thinking about our interaction with water. Yeah, so our polar molecules, they tend to have those oxygens in them, those hydroxyl groups. They're going to interact with that water. They're going to be attracted to the water. So because they're attracted to the water, they mix in and they dissolve. For our lipids, they're non-polar. And for this, we want to remember a principle of like dissolves like. So what we have dissolving in something non-polar is something else that's non-polar. So basically lipids would dissolve in lipids. Okay. So that brings us to enzymes. So enzymes are one of the many important roles that proteins play in the body. The enzyme acts as catalysts. So when we have a chemical reaction that's happening in the body, often, even if it's a technically spontaneous reaction, they don't happen at the speed that we want them to happen. We want to be able, first off, we want to be able to control the pace of chemical reactions, but also we want them to happen faster than, if you just sit a bowl of molecules on the table and tell them to do their thing. So enzymes are going to let us do that. So the way they do it is by decreasing activation energy for chemical reactions. So if we have a chemical reaction, right? Our chemical reaction, the basic part, right, is that we have something going into it, right? In a normal chemical reaction, we think about this as our reactant. We're creating a product. But when we add an enzyme, what we're doing is we are adding in the enzyme. It's going to bind on to our reactant. We're going to call that reactant our substrate. And then for a certain period of time, they're bound together. So this ES is like our enzyme substrate complex. Then when we get the product out, the enzyme breaks off again. And the enzyme is able to be reused at this point. The enzyme doesn't get used up. It's just lending a helping hand. We think about an activation energy barrier. Hopefully, you kind of remember this from chemistry, right? So what we have happening is we're going to have our reactants with a certain amount of energy. There's basically a hump of energy that they need to reach before the reaction then proceeds and we get a product. So what our enzymes do is they lower the activation energy. So the activation energy here, like this part, right? That hump is the activation energy. So our enzyme isn't going to change what our reactants start with. It's not going to change what our product ends with. What it changes is how much energy they need to accumulate in order to get there. So this is how they're able to speed up those reactions. Here we got it prettier with labels, right? So without an enzyme, we need a lot of energy. Right? So here we have our without an enzyme. I got this big activation energy. With our enzyme, much smaller. Our reaction coordinate is just as we're proceeding with the reaction in this direction. There are two sort of models, two ways people try to conceptualize how enzymes work. And the lock and key model is probably the one you learned if you thought about enzymes in like high school. So the way the lock and key model works is you basically think about our enzyme, right, as having a very specific shape at the site where it binds our substrate. Right, so we'd have a square, and we think about our substrate, our reactant, as being perfect. Go kind of deeper into our study of enzymes, into our study of chemical reactions in the body. It's not quite that simple. So this induced fit model actually explains things a little better. So enzyme reactions are often reversible. So like enzymes can do both the forward and the backward of these reactions. So the induced fit model basically just says that when our substrate binds onto the enzyme, we're going to have like a little bit of a shape change. So that induces, induce means like to produce, instigate, something like that. That's what an induced fit is. So we're basically saying that we kind of force things to fit and that it's not quite as specific as a lock and key. It's a little more general. Could bind a couple different things. So here we see this induced fit. We have our substrate. It's this circle, this S. Originally our active site, that's what we call the point where we bind on our substrate. It's kind of square looking, but our substrate is able to bind in anyway. Right? So that's that induced part. Right? There's a little bit of a shape change here as the substrate is binding in. Okay? And then our product is a different shape. It's released here. It just has to do with the fact that there's a shape change, really. Slightly better way of thinking about enzymes than the lock and key. Enzymes often need helper molecules, basically. So we have these two things called cofactors and coenzymes. The basic difference here is whether they're organic molecules or not. A cofactor is going to be something like a metal, so like an atom, a trace metal. So they're going to be non-protein components. They're like inorganic. The enzyme may need them to work, basically. So we'll see like iron plays a role in hemoglobin, for example. So that's a cofactor. A coenzyme, even though it has the word enzyme in it, it isn't an enzyme on its own, but it's an organic molecule that the enzyme needs in order to function properly. So kind of like a cofactor, it's just now we're talking about something more organic. So this is often derived from a vitamin. They're going to transfer small chemical groups onto the enzyme in order to allow it to do its job correctly. I'll give you a second with that. I see people are furiously taking notes right now. So finally, we want to think about enzyme reaction rates. So when we're thinking about enzymes, we know they're lowering the activation energy. Now that's speeding up a reaction, but there's more variation within that, right? Like we're not lowering every activation energy by the exact same numerical amount. And an enzyme can have different behaviors based on how much of the substrate, the reactant there is, how much of the enzyme there is, how attractive the enzyme is to the substrate. That in and of itself can vary. Right? You can have things that only sort of want to bind into an enzyme, or you can have things that are magnetized rapidly. Temperature and pH also make a difference because when things are at higher temperatures, they move around more, so that would like raise a reaction rate. pH kind of affects how things bind onto each other, because pH is basically about like how many hydrogens are floating around. Those are ions. Okay. So the catalytic rate, right? So you just make some definitions here, right? The catalytic rate is what we're talking about varying when we're thinking about enzyme reaction rates. So that's if we're counting how many molecules, so how much product is generated per unit of time. Like, so how many molecules per second are we making? And that is what is going to really be varying depending on these concentrations. So just to think a little bit more about this idea of concentration and this idea about affinity. So I'm going to draw to you what happens as we increase the amount of substrate. So why don't you tell me, if you have more of a reactant, do you think that reaction is going to happen faster or slower? So
Wait, you have more of a product?
more of the reactant, right? Yeah, say you're trying to get molecule B, you're trying to make it out of molecule A. Then you have lots and lots of molecule A all of a sudden, right? You started out with just maybe 1, now you have 5, now you have 100. It makes it easier to make molecule B just because sum of chemical reactions is dependent upon random chance. So that's generally what we call like the law of mass action, basically, is that if there's more of something, you get more of something. Okay. So our substrate concentration going up causes our reaction rate to go up. Now, what I've drawn here, we're going to think of that as something with low affinity. So we're saying here that this first line I drew was something that doesn't really want to bind into our enzyme that much, right? Like it's okay with the enzyme, but it doesn't love it. Now we want to think about what happens if we have a substrate that is super attracted to our enzyme. As we increase the substrate concentration, would I draw a line above or below that first one? So here's our, I'll label it, right? So here's our low affinity. That the substrate and the enzyme are more attracted to each other. Is it going to be easier or harder to get the product? Think about it like you're trying to get a group of friends together. Is it easier or harder if your friends really, really like each other? Or if it's like someone you vaguely know? Is it going to be easier to make that party happen or harder if they really, really like each other?
Easier.
Yeah, it's going to be easier, right? So if we have high affinity, our line's going to be above. You don't need as much substrate in order to make that reaction happen. These two things like each other, we get more product, we produce more results, more effect. So you can see we're actually a little bit over time, so we are going to end here. I'll stick around for questions, and I'll see you all Friday. Don't forget about mastering A&P.